Okay, welcome back. 5.2. Now we look at enzyme. This subtopic is very important. Why? Because enzyme is very important. So in a cell, we know that biochemical reactions, right, the metabolism that we said just now, it involves a lot of processes. So these reactions have to happen at a high rate, okay, to protect the living processes. So biochemical reactions can happen very fast because of these enzymes. So we know that this enzyme is something very helpful. So what is actually an enzyme? It is an organic catalyst, okay, that is mostly made up of proteins. You need to remember this point, yeah, proteins, and it is produced by the living organisms. So here is an enzyme where you can see it has active sites, and this is the substrate, and you can see that it fits nicely, it complements into the enzyme. Okay, however, not all enzymes are synthesized from proteins. Okay, these substances needed for an enzyme reactions are called substrates. But mostly enzymes are synthesized from proteins lah. The substances needed for an enzyme reactions are called substrates. So this is the one. So substrate will bind with the enzymes at a specific site. This is the active site. And when they are formed together, we call it as enzyme substrate complex. So there's a video here for you. This is the Okay, you can watch this video yourself later. This is called the lock and key hypothesis. So you can remember like this. You have the lock, okay, which is representing the enzyme, and you have the key, which is representing the substrate. So in the end, remember that the lock is still intact. Okay, it is still unchanged, it is still not consumed, but in the end, what will happen is that the key will break into two. All right. Okay, so next is on enzyme nomenclature. Enzyme nomenclature means this is the way the enzymes are being named lah. So very simple. This one you look at ASE. So the first naming of enzyme is like this. ASE is added to the substrate. Okay, added to the substrate. For example, lactose. So you change this to lactase, ASE. So lactase will catalyze these reactions of lactose, the hydrolysis. Hydro means water, lysis means breaks. So you can see here lactose, it will react with water. Water will, in the presence of water, this lactose will be broken into smaller products. Okay, so if you still remember in chapter 4, lactose is disaccharide, right? It's a disaccharide, okay, which is made up of two monosaccharides. These two are the simple sugar. Okay, however, there are also a few enzymes that do not follow this naming. Okay, this naming means the ASE lah. So some other examples are trypsin, pepsin, and rennin. Okay, especially enzymes were discovered before the systematic naming was introduced. So that means these enzymes are the earlier ones. Okay. Okay, next, this is a very important slide because you need to know the general characteristics of enzymes. Right, so we start with the first one over here. It says biological catalyst, speed up biochemical reactions. Okay, we already defined what is enzymes. Enzyme is organic catalyst. It is going to speed up, or it is going to make the reaction occur at a high rate. So this is first characteristics. Number two, it acts very fast, very quickly. So therefore, it can speed up the reaction. And number three, point three, it is required in small quantities. Okay, therefore it is reusable. Okay, next, the structure of the enzymes remain unchanged, or in other words, they are not destroyed after a reaction. Okay, like the lock and key just now, you still remember this enzyme is represented by the log. So in the end, what will happen to the log? You can still use the log again. It is not changed. It is not consumed. Okay, next, the reaction of enzymes are specific. So remember, substrate and the enzyme, they will bind to each other, forming enzyme substrate complement. Okay, so very specific. If it doesn't fit into it, okay, what will happen is that this enzyme is not going to catalyze the reaction of the substrate. Right? So specific is the keyword. Next one, most of the reactions are catalyzed by enzymes are reversible. If you look at the word equation, right, the arrows are double-sided, so that means these the reactions are reversible. Okay, next, inhibitors is the keyword. So by the name itself, inhibit, so it will serve to slow down or to stop the action of the enzyme, the enzyme activity. So example of inhibitor, heavy metal. Alright. Okay, next, some enzymes need cofactors to work more efficiently. Okay, an example of cofactor is vitamin B. It's like a helper lah. Right, so that's why you need to take some of this supplement, because this supplement is going to help the enzymes to work, okay, to speed up the biochemical reactions. Okay, next. Okay, extracellular, intracellular enzymes. By the word itself, intra, inside, extra, outside. So we have these two types of enzymes. Okay, this one very simple also. Enzymes that are synthesized in the cell and then it is used for its own use. It is called intracellular enzymes. So example, cellular respiration. Again, this is actually a very complex process, and in the series of cellular respiration, where glucose being the substrate, it is being hydrolyzed. So that means glucose is big, right? It is going to be broken down into smaller substances. So they need this hexokinase enzyme. Okay, you know glucose 6 carbon, so that's why the name hexo. Okay, so this is an example of intracellular enzymes. On the other hand, we have also extracellular enzymes. So maybe you are also familiar with this name, trypsin enzyme. Okay, trypsin enzyme is produced by the pancreatic cells, okay, but it does not work in the pancreatic cells. It is then secreted into the duodenum. Do you still remember what is the duodenum? Which part? It is actually the first part of the small intestine. So meaning it is produced by pancreatic cells, but then it does not work in the pancreatic cells. It is being secreted out into the duodenum, and in duodenum, what does it do? This trypsin enzyme will break down polypeptides. So that means protein hydrolysis. Protein is a very complex molecules, but then it is broken down into smaller chains of polypeptides. Okay, until eventually it is going to be broken down into amino acids, right? We have a lot of these enzymes involved. Okay, pepsin, trypsin. Okay, next. This is in the syllabus where you need to know these extracellular enzymes. How are these extracellular enzymes being produced? Okay, so we stopped here.